Cell structure and homeostasis. For this biology praxis prep, we will start with a summary of cell structure and homeostasis and follow that with some practice questions similar to what you might see on your praxis exam. Cell structure and homeostasis. Prokaryotes versus eukaryotes. Eukaryotic cells are usually larger than prokaryotic cells. Prokaryotic cells have cell walls made out of peptidoglycan, a polysaccharide containing sugars and amino acids, while eukaryotic cells have cellulose cell walls, plants. Both eukaryotes and some prokaryotes have a phospholipid bilayer with proteins and cholesterol membrane. Eukaryotic cells have small membrane-bound organelles such as mitochondria, chloroplasts, ribosomes, and the nucleus. The eukaryotic nucleus houses DNA. Prokaryotes do not have a nucleus. Their DNA is carried on a small circular chromosome in the cytoplasm. Eukaryotes have linear chromosomes. Eukaryotic cell structure. Nucleus. The control center of the cell houses and protects the DNA, assembles ribosomes in the nucleolus, copies DNA instruction into RNA via transcription. The endomembrane system produces and transports the cell's proteins and other products or wastes, includes the nuclear membrane, endoplasmic reticulum, Golgi apparatus, transport vesicles, cell membrane, and lysosomes. The Golgi apparatus selects and sorts chemicals in the cytoplasm to be moved to lysosomes or dumped out of the cell. Lysosomes break down polymers into monomers that are recycled in the cell. They are not present in plants. Ribosomes are made up of ribosomal RNA, rRNA, and proteins. Translation occurs when mRNA directs the ribosome to make proteins. Cell or plasma membrane is a phospholipid bilayer with proteins and other molecules interspersed throughout. It is the barrier between the cell interior and the external environment. Cell walls are found in plant cells only. It's a rigid wall made of cellulose protecting the cell interior from the external environment. Cytoskeleton is made of long protein tubes and fibers that guide the internal movement of organelles and macromolecules. Animal cells have a more extensive dynamic cytoskeleton than other cell types, which helps compensate for their lack of a cell wall and gives them motility or movement. Chloroplasts only reside in plant cells and some prokaryotes. They convert solar energy into energy stored in the cell via photosynthesis. Mitochondria convert chemical energy from glucose into ATP during cellular respiration. Vacuoles are membranous sacs that come in different shapes and can store food, chemicals, pigments, and waste products. Central vacuoles in plants store water to make the cell stronger and more rigid. Selective permeability and transport. Membranes allow for the intake of nutrients and the elimination of waste because they are selectively permeable. This means they can let some molecules pass through and not others, maintaining a separate internal environment. Aqueous solutions are typically found on both sides of any cell membrane, internal or external. Membrane transport involves the movement of solutes or water across a cell membrane. Cells maintain homeostasis and do cellular work with membrane transport. Passive transport requires no input of free energy, while active transport requires an input of free energy. You can see in this membrane image here the allowance or passage of these green-blue particles, while the yellow must remain on one side. Passive transport, the movement of molecules, ions, solutes, and water through a cell membrane without the input of free energy. It occurs where the concentration of a substance is greater on one side of the membrane than the other. the particles move with the concentration gradient. In diffusion, the random movement of particles in a gas or liquid from an area of high concentration to low concentration. In osmosis, the net diffusion or flow of water molecules through a semi-permeable membrane into an area of higher solute concentration. And facilitated diffusion, which is similar to diffusion, but requires the help of an integral protein in the membrane. Diffusion. Membranes often act as the barrier between areas of high and low concentration of a solute. Solute molecules diffuse in all directions, but the net movement is from areas of high concentration to areas of low concentration. Molecules that are allowed to cross a membrane will diffuse until their concentration is at equilibrium, equal on both sides of the membrane. So we start with this high concentration in the extracellular space outside of the cell, and we get this diffusion from high concentration to low concentration until we reach equilibrium. Facilitated diffusion. Small molecules like oxygen and carbon dioxide readily diffuse through all cell membranes because they are small and nonpolar. They can squeeze between the phospholipids. Larger molecules, polar molecules, and ions cannot squeeze between the phospholipids, or they're repelled by the hydrophobic membrane interior. They need the help of a transport protein. As in passive diffusion, particles undergoing facilitated diffusion move from an area of high to low concentration of solute. Unlike passive diffusion, particles cross the membrane with the help of an integral protein. Since the substances are going with their concentration gradient, this is a type of passive transport. No energy is needed. Types of passive transport proteins. Channel proteins provide corridors that allow a specific molecular ion to cross the membrane. Aquaporins are an example. Carrier proteins bind to a specific solute and change shape, helping it move across the membrane. Proteins in the cell membrane. There are 3 types of proteins in the cell membrane. Peripheral proteins, like you see labeled in orange here, are not embedded in the membrane, but are anchored on one side. Integral proteins are embedded in the membrane. So you can see there are 2 types of integral proteins, monotopic or transmembrane, labeled in red. Transmembrane proteins are a type of integral protein that span the entire bilayer from one side to the other. Cell signaling molecules pass through these proteins. Active transport. Active transport uses energy to move solutes through a transport protein against their concentration gradients. It takes energy to paddle a boat against the current, but no energy to float with the current. Active transport moves against the current. The source of free energy needed is often ATP, which provides energy for cell processes. Active transport proteins may move 1 or 2 solutes in either direction or opposite directions across a membrane. So the star here is the energy provided via ATP, and we can move a solute in one direction. It could move the solutes in both directions or in opposite directions across a membrane. The sodium-potassium pump, example of an active transport protein. The sodium-potassium pump is found in the plasma membranes of all animal cells. It uses the energy of ATP to pump sodium and potassium in opposite directions against their concentration gradients. Water potential and osmosis. Water potential is a measure of the concentration of free water in a given solution. So if you look on the left here, we have a low water potential on this side and a higher water potential on this side. Osmosis is the net diffusion or flow of water molecules through a semi-permeable membrane, so you see the semi-permeable membrane here, into an area of lower water concentration. So water flows from higher water potential to lower water potential. This difference in concentration is called the concentration gradient. A solution has a higher water potential when it has a higher water concentration and lower solute concentration, as you see on the left here. When water potentials in neighboring compartments are compared, the direction of osmosis can be predicted. Water flows from areas of high water potential to areas of low water potential. Therefore, water flows from areas of lower solute concentration to areas of higher solute concentration. Let's look at some conditions both inside and outside the cell and talk about what the net direction of the water would be, or what the net direction of the osmosis would be. So, if the outside solution, so if outside of the cell is hypertonic, then the greater solute concentration is outside of the cell, and the greater water concentration is inside of the cell. The net diffusion of the water, the net direction of the water diffusion will be from inside to outside. So from the greater water concentration away. And what we would see is this flow of water from inside to out. If the outside of the solution, so if the solution outside of the cell is hypotonic, that means that the greater solute concentration is inside the cell and the greater water concentration is outside. So the water will flow from out to in. So we see the net flow of water into the cell. If the solution outside of the cell is isotonic, that means the greater solute concentration is the same. It's the same both inside and outside. The water concentration is the same both inside and outside. And the net direction of the water diffusion would be none. So we'd have this equilibrium, this dynamic equilibrium from in to out and out to in of the water. Cell signaling. All living things respond to their environment. All living things alter their environment in ways that affect other living things. They do this by sending and responding to signals, a characteristic of living things known as cell signaling. Signals and responses occur at every level of organization, from the biomolecule to the biosphere. Cell-level signals and responses from cell to cell, from within the cell, and from the environment. Signal and response. Multicellular organisms coordinate the activities of their cells, tissues, organs, and organ systems by sending signals between cells both near and far. Cells in multicellular organisms are specialized, genetically limited to respond to certain signals in their environments and ignore others. Cells may receive multiple signals at any given moment from both its internal and external environment. The signals a cell receives will vary as its environments vary and as the needs of the organism varies. The combination of signals that a cell receives at any moment, as well as its specialized function, will determine how it responds. Types of cell signaling. The types of cell signaling are defined by how the signal molecule or ligand is received. Autocrine is where the ligand is received by the same cell that sent it. Juxtacrine is where the ligand passes directly into an adjacent cell. Paracrine is where the ligand is received by a receptor of a nearby cell. And endocrine is when a ligand is received by a receptor of a distant cell. Cell responses. Cellular responses to a particular set of signals could potentially include everything cells do, such as activation of cytoplasmic enzymes, change in gene expression, entry into the cell cycle, pause in the cell cycle to repair DNA or organelles, apoptosis or programmed cell death, maturation or specialization of cells, and movement, migration, or shape changes of cells. Internal signaling. Critical signals also come from the cell's internal environment and don't involve cell surface receptors. For example, damaged chromosomes lead to signals that cause cells to pause and repair. Internal signals and responses are crucial for the maintenance of homeostasis. Exocytosis. To excrete a macromolecule from the cell, the vesicles that enclose the proteins fuse with the plasma membrane, and the vesicles then open up and spill their contents outside of the cell. This process is known as exocytosis. The vesicle will become part of the membrane. This is how secretory proteins from the Golgi exit the cell, and is true for insulin in the pancreas as well. Endocytosis. The opposite of exocytosis is endocytosis. In this process, the cell takes in macromolecules or other particles by forming vesicles or vacuoles from its plasma membrane. This is how many protists ingest food particles. Let's look closer at some forms of endocytosis. Phagocytosis, illustrated here, takes in solid particles. Pinocytosis, indicated here, takes in liquids. Now, the requirement here isn't for the intake of the liquids, but for the substances that are dissolved in the liquid. Receptor-mediated endocytosis, illustrated here, requires the help of a protein coat and receptor on the membrane to get through. Feedback loops. A feedback loop is the path that leads from the initial generation of a signal to the modification of an event. Feedback loops can be positive or negative. A positive feedback loop is where a signal is continually amplified until a desired outcome is reached. The key to these loops is that any small change will be amplified. A negative feedback loop occurs when the outcome of action reverses the cause of the original signal. A negative feedback loop is used to maintain a stable blood glucose level, which is a crucial type of homeostasis. When blood glucose is low, the pancreas releases glucagon, a peptide hormone, which induces the liver to break down stored glycogen and release glucose into the blood. When blood glucose is high, the pancreas produces insulin, which causes liver cells to take up glucose from the blood and build glycogen molecules for storage. The inflammatory response is an example of positive feedback. When injury or infection occurs, histamines, small signal molecules, are released at the site of damage. Histamines cause local blood vessels to dilate. Blood flow is increased, which causes redness and swelling. The redness and swelling, in turn, causes more histamines to be released. The cycle restarts and is continually amplified until all pathogens are destroyed. When that signal is removed, the positive feedback loop stops and homeostasis is restored.